And so this is when you have a mismatched base pair, and that's what's shown with this bulge here because it doesn't have the proper hydrogen bonding. And the E. coli has to figure out which half of the mismatch is incorrect, and it makes its decision based on the fact that older DNA, which is assumed to be the correct DNA, gets methylated, and so that it will remove the nucleotides on the new strand and then synthesize it. Okay, so that's mismatch repair. Another mechanism for DNA repair is base excision repair. And this is when the actual, it starts out with just a damaged base that is removed by a glycosylase. Okay, so remember, so this is going to be, we have our ribose just schematically, and then you have the covalent bond between the ribose and the nitrogenous base. The glycosylase breaks that bond, right? Because that is a glycosidic linkage because it's between a sugar, that's where the glyco comes in, and the base. And that's what's represented here. So notice in this, the glycosylase takes out the base, the backbone is intact. This is a distinction between this mechanism and nucleotide excision repair, is that the first step is to create an AP site. That AP stands for apurinic or apyrimidinic, meaning it's just the base that is removed, but still the backbone, the sugar and the phosphates are intact. Then an endonuclease, let me see. Yeah, you get an excision exonuclease, or called an exonuclease, takes out more nucleotides, and then DNA polymerase 1 comes in and refills. And then remember that ligase is the enzyme that can join, that can seal nicks in the backbone. Okay, so what is it, like 4 steps? Glycosylase, exonuclease, then putting it back, and then ligating. Okay, so that is one kind of repair. And then another kind is nucleotide, and actually, this is often abbreviated BER, base excision repair. And then nucleotide excision repair is abbreviated NER. And this is often a repair mechanism for UV damage. And again, DNA polymerase 1 and DNA ligase are involved in the mechanism. So you have some kind of insult to the DNA, as I mentioned, often caused by UV damage. And that's what's represented here. And in this case, instead of just taking out that base, it takes out a whole big piece of DNA surrounding it. And then DNA polymerase 1 and ligase comes in and puts it back. So if there is a double-stranded break in the DNA, you can imagine that that is a more dangerous situation and poses a greater threat to the organism. And there is this process called recombination, or non-homologous DNA end joining. And I don't talk too much about this because I don't fully understand actually how that works. But that's another mechanism of repair. And recombination is something that's usually covered a lot in biology. Kelly, did you have that in your microbiome? It was micro that you saw this stuff in, right?
Yeah.
They spend a lot of time on the holiday junction.
Not that I recall.
Okay, so recombination is this way of taking one strand from one section of DNA and another strand, and they cross over. And so you get swapping of strands. And I've never spent a whole lot of time. I've actually done it in the lab. We had an experiment in the biochem lab I taught at USD. I had someone who also taught with me that did recombination in her PhD project, and we had a little model you could do. But I don't fully understand how it works in terms of drawing all the strands, migrations. But I did just learn, I saw this and I was trying to find more details on it. There's this new COVID Omicron variant called XE. And apparently XE is a recombination of BA1 and BA2. It's not just a simple mutation, which usually results in an error in the copying. It's a recombination between two different strains. I sent myself the TikTok where it was described the difference between the recombination and the mutant, and I can't find it. It was a really good description though. So this is what the recombination, the cartoon of what it looks like is. But there's this branch migration, and it's a possible mechanism for repair. That's all I'm going to say about that. Okay, now you can imagine pretty much everything we've looked at has been in E. coli, and you can imagine that replication of DNA in eukaryotic systems is going to be more complicated. First of all, the genome in E. coli, we call it naked DNA because it's not packaged in chromatin. But in eukaryotic systems, we have histones and those nucleosome beads on the string. And so those all have to be managed at the same time DNA is replicated. And also there is this growth cycle for copying cells in eukaryotic systems. And the growth cycle is described by the 3 different phases: mitosis, where the cell division occurs, the G1 growth phase, and then the S phase where DNA replication takes place, and then the second growth phase in preparation for cell division, and then you get to cell division again. This is called the cell cycle, and the cell cycle is of a lot of interest because most of us are probably aware that cancer is the result of unregulated cell replication, right? Because cells are dividing too fast, and then you get tumors which need nutrients and blood, and so they take over things that are necessary for healthy tissue. And so some elements of the cell cycle have been related to cancer, when there's dysregulation of it. It's been one of the targets for cancer therapeutics. Okay, so in eukaryotic replication, remember that there's long genomes, and so there's multiple initiation sites for the origin of replication, whereas in E. coli, there's just the one. And remember, the site in E. coli where it begins is called OREC. This is DNA in E. coli. Oops, I'm getting notifications. Oops, there. The complex that is the assembly of all the proteins necessary for replication is called the origin recognition complex. Sorry. Then there's a replication activator protein, and then replication licensing factors. Cyclins are a class of proteins that are associated with the cell cycle. And then cyclin-dependent kinases. Remember, kinases are proteins that phosphorylate with ATP. Those are all involved in replication. I'm not too worried about this. I think I would rather have you focus on the aspects of bacterial replication. So I'll say you can skip this. Okay, what I do want you to be able to look at critically, though, is all the different polymerases that are in eukaryotic systems. So, so it says there's at least 19, imagine that, different DNA polymerases in eukaryotes. 5 have been studied more extensively, right? So we have alpha, delta, epsilon, beta, and gamma. And remember, we were looking at a couple properties of DNA polymerases in terms of identifying the ones that would be the polymerases responsible for copying the genome. So if we look at the data here, we can see that, okay, gamma is found in the mitochondria. So that one's going to be responsible for copying mitochondrial DNA. So that one's definitely not going to be the one that copies the genome. Then there's processivity. And do we have a KCAT? I don't think they gave us a KCAT here, which is kind of a bummer. Properties, processivity. All right, so in terms of processivity, though, what do we want, low or high processivity for the one that copies the genome? Yeah, we want, so it's only going to be, and unfortunately, on this table, they don't give us actual numbers, but we're going to be looking for a very highly processive DNA polymerase, right? So we have the ones that are highly processive are delta and epsilon. So it's going to be one of those two. And then we also need high fidelity. So fidelity that's a term used to describe error-free. So we obviously want a DNA polymerase that's going to copy the genome to have very high fidelity. And notice that, okay, that most of them have 3' to 5' exonuclease activity. So I'd say looking at these two, delta and epsilon, those are the two that are most likely going to be the ones that copy the genome. And I believe it's delta is the one that is has the most processivity. What they don't mention here is pCNA. And then one of these is a primase, that's alpha. So that means that if it has primase activity, that means it can lay down bases without the 3' hydroxyl. So DNA polymerase alpha is the one that is responsible for putting down primers. And delta is the one that copies the genome. And I think epsilon is thought to be involved in repair.
Can I ask a question about these polymerases specific to the eukaryotes? So are we the Greek alphabet to label them? Like, is that an indication? Okay, this is polymerase just for eukaryotes and not for like E. coli? Because the E. coli was like 1, 2, 3, and A, B, C.
So yes. So if you see alpha, delta, epsilon, beta, gamma, they are talking about eukaryotic polymerases. 1, 2, and 3 are the bacterial. But of course, like, it's never that simple. So if you look at yeast, they have different names. And for every organism, there is the potential for the polymerase to have a different name. But then what they do is they look at the properties and the sequence homology and try to determine which of these it's most similar to: alpha, delta, epsilon, beta, and gamma. Does that make sense?
Yeah, thank you.
It makes reading stuff like it makes reading literature very challenging because if you venture into a new organism, then you have to figure out what's what. Again, most people work within one organism, right? And so they just get used to thinking of it that way. Okay, the thing I want to point out here is pCNA, proliferating cell nuclear antigen. Whenever you find a DNA polymerase that is found to be in association with a pCNA analog, meaning something very similar to it in a different organism, that is the polymerase that's responsible for copying the mechanism. I mentioned the sliding clamp in E. coli, the beta subunit. That is the subunit of DNA polymerase 3 that increases the processivity and makes DNA polymerase 3 really efficient at copying the E. coli genome. The eukaryotic analog to that is pCNA, which is funny. You learn terms and then you don't really think about what they individually mean, and this name is probably a result of how it was described or how it was discovered. So, right, proliferating cell, meaning a cell that's going to be dividing, right? Okay, so a cell that's dividing is going to need DNA polymerases and their associated and the things that are required for DNA replication. So this was found in proliferating cells in the nucleus, and I guess this is it. It was an antigen, so they must have used antibodies to find it. So it's a strange name, but what you will see in the literature, if you're looking at DNA polymerases in different organisms, they will say, oh, this one is associated with a pCNA-like protein. And they use that as evidence to figure out it's the protein that is involved in replicating the genome. Epsilon is involved in leading strand replication and also can replace delta in lagging strand synthesis. Beta is a repair, gamma is in the mitochondria. Okay, and this is showing just the structure of pCNA and how it's circular around the DNA, just like the beta subunit. And this is a comparison of the polymerases in bacteria and the polymerases in eukaryotes. So one origin of replication, several origins of replications. In this one, in bacteria, all polymerases have exonuclease activity. I think alpha does not in the eukaryotic, and there might be another one. The Okazaki fragments, remember, those are the pieces of DNA that are made in lagging synthesis. They're actually longer in bacteria than they are in eukaryotic systems. And then the other major difference is that there's no proteins in complex with the bacterial DNA, but the eukaryotic systems have histones. And so those have to be unpackaged for replication to take place. So eukaryotic replication fork. And they're showing topoisomerase to relieve the positive supercoiling upstream of the replication fork. Helicases, which are again responsible for strand separation. RPA is probably similar to SSB in terms of keeping the single-stranded DNA apart and safe. Remember, ligase is the protein that joins Okazaki fragments. RNase H. This is a protein that it removes the RNA primers. Do you guys remember here's a brain check. What removes the RNA primers in prokaryotic replication? Anybody remember? Right, we have at the beginning of each Okazaki fragment, there is an RNA primer, and that has to be removed. It's a polymerase. Do you remember which one?
Is it 3?
3 is the one that copies the genome. There's only one that has 5' exonuclease activity. It's DNA polymerase 1. Uses its 5' exonuclease activity to remove the primers. Random fact, just meant to jog your memory. Oh, and then this is just to mention that there are telomeres at the end of your DNA sequences. Have you guys heard of telomeres? These long stretches of repeated DNA sequences at the end of chromosomes, and it requires a special protein to copy them. And every time a cell undergoes cell division, the telomeres get shorter. So it's one of the, it's actually a hallmark of aging, is when you are young, you have very long telomeres. And I think it's thought to be protective of the ends of the chromosomes. But every time you go through cell division, a cell goes through cell division, the telomeres get shorter.
Professor?
Yeah.
Is that only in eukaryotic DNA?
Yes.
Okay.
Yes. Yeah. And I think they didn't know. So I remember they found the sequences, and then they, synthesized them and looked at what they did in the test tube. They make these sort of like tetrameric ends. It's a different DNA structure. They didn't understand the significance of it until later. Okay.
Why don't prokaryotes have it?
Good question. Probably too much of an investment metabolically. And right, so bacteria, the DNA in, say, our brains needs to be in our neurons in our brain need to be protected for 80 to 100 years, because some of the neurons don't continually divide. In bigger organisms, cell division happens much less often than in single cellular organisms. So E. coli live 20 minutes. Well, they don't, their generation time is 20 minutes. And so they don't need to invest a lot of energy into protecting the genome, because they're just going to have daughter cells, or the next generation comes along. But in our bodies, some cells have to last decades and decades and decades, and so we really need them to be protected.
Okay. I'm also looking at the homework, and it's asking about telomerases and what would happen if they were inactivated. So can you repeat again, like, what they do when they are activated?
So to tell her, let me pull up what I was thinking in terms of that. Hold on. This is chapter 10. There we go. What question number is that? It's at the end, right?
Yeah, it's 56. I was also going to ask if we can also go over 55.
Okay, so what would happen to DNA synthesis if telomerases were inactivated? So it would eventually stop DNA synthesis. So the telomerases ensure that there's adequate ends at the 3' end of the strands so that a primer can be placed. And so the ends would just keep getting shorter, and then there'd be no room for a primer. Okay. And replication can't happen if there's no primer.
Gotcha. Thank you.
Okay. Okay, now in terms of for 55, DNA synthesis in eukaryotes and stages of the cell cycle. So I would list the different phases, G1, S, G2, and M. S is when this replication takes place, DNA synthesis occurs. G1 is preparation for synthesis. And so there are checkpoints in G1 to make sure that everything's ready before the DNA is copied. And then G2 is the second growth phase. So DNA synthesis is done, but the cell continues to grow. So it's probably prepping for breaking the cell membrane and making organelles. And there's a checkpoint there to make sure that there's regulation there to make sure that everything's ready before mitosis in the M phase.
Okay, thank you.
Okay. Oops. Okay, so that's the end of replication. Then there's transcription. And you guys still see that, right? See the PowerPoints.
Yes.
Okay. Okay, so basically, in all of our cells in our bodies, the genetic makeup is the same. We have the same DNA in all the different cells, but the cells look different. And that's because different parts of the genome are expressed in each cell. And that process is transcription. Let's see, transcription is when the DNA is expressed and messenger RNA is made, and that goes on to create proteins in translation. Okay, so this is when RNA is synthesized, and so we need all 4 of the RNA nucleotides. So instead of the deoxy ATP, now we need just regular ATP, GTP, CTP. Instead of thymidine, now it's uracil. And whenever you see nucleotides, think magnesium. One big difference between RNA synthesis and DNA synthesis is that a primer is not needed for RNA synthesis. So RNA polymerases do not have a requirement for that 3' hydroxyl, but they do need a DNA template to know what sequence to make, which I think would be pretty intuitive. Just like DNA polymerases, synthesis goes 5' to 3', which means that the template will be 3' to 5'. And the template is unchanged. So this is a cartoon showing what the sequences would be like. So here we have the DNA, the genetic portion, and there is what's known as the coding strand and the template strand. So notice our template strand is 3' on this end, and 5' would be down this end. And the bases that are in the messenger RNA are going to be complementary. So T to A to U, C to G. The RNA transcript is going to be complementary to the template strand. There's a lot of nomenclature for this. Because it's complementary, this would be called the minus strand, and the coding strand is going to be the same sequence except for T to U as the messenger RNA, right? So if we look at the 5', it's going to be the same. ATG, this is AUG, because T gets substituted with a U in the messenger RNA. The coding strand is also often called the sense strand, and the template strand is called the antisense strand. The messenger RNA goes on to code for the amino acid sequence in proteins during translation. I have a video, where did I link it? I think this video goes over the nomenclature and sequence for the coding and template strands. Let's skip there. Okay, so in E. coli, there is just a single RNA polymerase that catalyzes the synthesis of the messenger RNA. It's pretty big. It's almost 500,000 in molecular weight. And it has a pretty complex subunit structure. There's alpha, omega, beta, beta prime, and then sigma. The RNA polymerase without the sigma subunit is called the core enzyme. And when it has the sigma subunit, it's called the hollow enzyme. So the core enzyme is capable of synthesizing the messenger RNA, and it does it faster than in the presence of the sigma subunit. The sigma subunit in the hollow enzyme, this is key for directing the RNA polymerase where to go on the genome. Okay, that's a lot of what I was talking about. The template strand is called antisense, and it's the minus strand. And the coding strand is the sense strand, and it has the same sequence as the messenger RNA. The sigma subunit recognizes the promoter. The promoter is a sequence of DNA to which RNA polymerase binds in order for transcription to begin. Okay, so, right, we have this whole big genome, and only some subset of it is going to be transcribed. And so the RNA polymerase needs to know where to bind on the DNA, and what it does so by binding promoter. That's that. That's basically what I just said here. Here's a video about where the promoter, about the promoter. Okay, let me see if I can get one. So a little bit of, well, I'll just do a schematic here. So let's say we have some sequence of DNA, and by definition, the start site of transcription is called +1. The genes, the genes that are being transcribed, those are called structural genes. These are things that will become proteins. Those are downstream. And they're numbered 1 to whatever, but they are called downstream. If you go in, I don't know how to say it without upstream. If you go in the other direction, it's called upstream of transcription. And upstream of transcription, there's 2 important regions, the -10 region and the -35 region. And those are parts of the promoter. And that's what's shown here. There we go. Okay, so +1 is almost always a purine. The -10 region, these are all different sequences of promoters in E. coli. So these are genes that are transcribed or operons, and these are the associated promoters. The -10 region is called the Pribnow box. And if you take them all and line them up, the most common nucleotide in each position in the Pribnow box, the one that's the most common is called the consensus. And so the consensus sequence for the Pribnow box is TATAAT. Okay, so notice that the Pribnow box has lots of A's and T's. So in my mind, that makes me think about one thing in the process of transcription. Any ideas what that could be when you see lots of A's and T's? Think 2 hydrogen bonds instead of 3 hydrogen bonds in the base pairs.
Does it have to do something with like uracil, since we're going from DNA to RNA? And
Well, there'll be, so that part is not transcribed. Transcription begins here and it goes that way.
oh, okay.
But GC base pairs have, so this is the DNA, it's double-stranded, and there's only 2 hydrogen bonds between the A's and the T's. You have to, in order to transcribe the DNA, it needs to be separated, the duplex. And so this is probably where the transcription bubble starts, because that region of DNA can be opened up more easily. Okay, so that's the Pribnow box. At -35, there's called the -35 region, and this is the consensus sequence here for that. Okay, so transcription is in E. coli, it's very highly regulated by RNA polymerase binding. And once RNA polymerase binds, you have this complex that is called the closed complex. But transcription requires the open complex, which is the opening of the strands. I'm going to show it here. Okay, so here we have E. coli RNA polymerase. It's shown as having that sigma subunit. So remember, this part is the core enzyme. Everything together is the hollow enzyme. The promoter is a region of DNA where the RNA polymerase binds before transcription begins. So this is going to be, between -10, -50. And so transcription start would be somewhere here. This is the closed complex. The DNA strands separate, you get the open complex. And then the first base is usually a purine, so an A or a G, and that's the first nuclear NTP to come in, and then you get synthesis. Once things begin, that sigma subunit diffuses away from the hollow enzyme of RNA polymerase, and then transcription takes place. And the process of synthesis we call elongation. For each of these processes, it's helpful to create kind of a table where you think of how does it start, initiation, what happens during elongation, and then how does it end? Because you're going to find that between reading about prokaryotic and eukaryotic systems, replication, transcription, and translation, it starts to get all modeled. But if you can organize it on a table, it helps to keep it helps you to understand things more. So this figure is showing that there are 2 possible ways that that elongation of the messenger RNA could take place. One is that the RNA just polymerase winds around the double helix, or that the RNA polymerase unzips the double helix, and that's how it goes. And that's generally to be accepted what's going on. So notice, okay, here's our messenger RNA that's being synthesized. Here's the transcription bubble. That's what we call the open complex. And when the RNA polymerase is unzipping the duplex DNA, that's going to cause positive supercoils upstream here. And so there's a topoisomerase that removes the positive supercoils. And then downstream of it, there will be negative supercoils. And so there's a topoisomerase that removes the negative supercoils. Skip that. In terms of termination of transcription, there's two different ways, and they're both associated with having hairpins. There's what's known as intrinsic termination. And so it's not dependent on any proteins. It's just controlled by the sequence in the messenger RNA. So what happens is in the transcript, this is showing the DNA sequence here, and it's showing that this sequence here, if you look at it, is complementary to this sequence. And when you make the messenger RNA, here's the A's are here, and then they're here, and these will be U's, they're here. What you wind up with is a single-stranded piece of messenger RNA that is self-complementary. So, right, because this is a single strand, and this region is complementary to that end, and that is how you get a hairpin. And certain hairpins decrease the stability of the RNA polymerase being bound to the double-stranded DNA, and then you get the end of transcription. So there's intrinsic termination, and that is based on this hairpin formation. There's also a type of termination called row dependent termination, but there's also hairpins involved in that, although they're not showing it here. So in row dependent transcription termination, there's this protein. That's showing a hexamer of row. Row is written like that. And essentially what it's thought of is that it chases the messenger, it chases RNA polymerase on the messenger RNA. So see, it's here and it's getting closer to the RNA polymerase, and then eventually it catches up to it. And in the versions of textbooks that I've read, there's also a hairpin, but it's not showing that here. But when it catches up to the RNA polymerase, it causes it to dissociate from the template DNA, and then you get the end of transcription. Okay, so there's a lot of ways to control transcription in eukaryotes, and even more in prokaryotes, and even more in eukaryotes, you can imagine. One of the simplest ways is there are alternative sigma factors. Okay, so remember, this is an RNA polymerase subunit. And this is the one that directs the RNA polymerase to the promoter. And remember that promoter, that is a piece of DNA upstream of a gene, a set of genes that's going to be transcribed. So it turns out, well, I think it will show next. It turns out there are different sigma factors. There's enhancers, which promote transcription. There are operons, which are collections of genes that are transcribed together or not. And then there's transcription attenuation, where transcription of a set of genes starts, but then gets terminated early. So these sigma factors, there are different ones depending upon, that will associate with the RNA polymerase, depending upon the situation. So for example, in E. coli, there is a sigma factor called sigma 70, and it gets made when the E. coli is exposed to heat. It's a heat shock protein. And so you can imagine that if E. coli is undergoing heat shock, it might need to start synthesizing different proteins and different genes. And so what happens is the sigma subunit gets made when the E. coli, and this is a protein, when the E. coli gets exposed to heat, then it associates with the RNA polymerase, and it delivers the RNA polymerase to the genes that need to get made when the E. coli is under heat stress. And so in prokaryotes, binding of RNA polymerase to the promoter is the rate limiting step for transcription. So if you have something that can deliver the RNA polymerase to a specific set of genes, that will be an effective way of initiating transcription. Okay. Then there are enhancers. So just to keep track, right? That sigma subunit, this is a protein component of RNA polymerase. Enhancers are sequences of DNA that bind transcription factors. A lot of vocabulary. So a transcription factor is a protein that associates with DNA and alters levels of transcription. So any protein that affects transcription is called a transcription factor. So some E. coli genes have sequences that are upstream of the promoter region. So remember, promoters DNA, enhancers are DNA, transcription factors are protein. And the genes that are involved in ribosomal RNA synthesis have enhancers called Fis sites. And those Fis sites bind a protein called Fis for activating transcription. Okay, so we're looking at the bacterial promoter for ribosomal RNA. Here is our start of transcription at +1. This is the promoter, the PRIBNOW box in the -35 region. Then there's an up element, which is an enhancer, and then the Fis sites also. Okay. So we've had alternate sigma factors, enhancers, and now there are operons. So an operon is, there's so much vocabulary. It's a collection of genes that are regulated and transcribed together, and it includes the regulatory elements. So there is the operator. Well, actually, let me start with what you've heard of. There is the promoter, right? That's that PRIBNOW box in the -35 region. And there are structural genes. Those are the genes for the proteins that are going to be made. There's a lot of DNA that gets transcribed into RNA that doesn't eventually become translated. And you'll see that they used to not know what they do, but now they know lots of them are involved in regulation of transcription and translation. And then there's also in the operon, there is operator DNA. These are the sites that transcription factors bind to. And these are generally pretty close to the structural genes, but you'll see in eukaryotic systems, they can be thousands of bases away. In bacteria, they're generally closer. So operons are not transcribed all the time. If something is transcribed all the time, we say it's constitutively expressed. So constitutive genes always expressed. An operon would not be constitutive. And so they're regulated by induction. Induction is a process where usually it's a small molecule triggers the expression of an operon. And I'm going to show you an example. And the molecule that does the induction is called the inducer. In biotechnology, this is often IPTG. It's a lactose analog. Okay, so they're going to talk about the lac operon. And this is a collection of structural genes for enzymes that degrade lactose. And so the structural genes are LAC-Y, that's the name of the gene, and it codes for the protein lactose permease. Beta-galactosidase is the LAC-Z gene. LAC-A codes for transacetylase. And LAC-I encodes for a repressor protein. So a repressor is a type of transcription factor that blocks transcription. So it's a protein. Okay, this is, I'm just gonna, operator, that is the sequence of DNA to which the repressor binds. I'm gonna show you the cartoon because I think it's easier when you see the cartoon. Okay. So this is a cartoon of the lac operon. We have the enzymes, the structural genes here. We have this is the gene for the repressor protein. O stands for operator. So remember, an operator is a piece of DNA to which a transcription factor binds. And this P tag here, that's the promoter. And the promoter and the operator can overlap, and they do here. So remember, just to review, I know I'm repeating stuff a lot, but I think it probably helps. The promoter is the sequence of DNA to which RNA polymerase binds. The operator is the sequence of DNA to which the repressor protein binds. If the repressor is on the operator, RNA polymerase can no longer bind to the promoter, and it blocks transcription of the genes. Okay, so what this is showing here, this is the gene that codes for the repressor. So this is the LAC repressor messenger RNA. It gets translated into its protein and it forms a tetramer. So it has oligomeric structure. And under normal conditions, the repressor binds to the operator. When the repressor is bound to the operator, RNA polymerase is blocked. And remember, the rate determining step for transcription of a set of genes is RNA polymerase binding to the promoter. So if RNA polymerase does not have access to the promoter, these genes are not transcribed. Okay. But you can have conditions where an inducer turns on transcription of the genes, and it does so by inactivating the repressor. Okay, so again, same structural genes. We have our repressor here, but now there's this inducer molecule. And in the cell, it would be allolactose, which is related to lactose. These are the enzymes that break down lactose. So if we have lactose in the cell, we need these to be made so that lactose can be utilized. And so this naturally occurring is allolactose, and it has a binding affinity for the repressor. When the repressor is bound to allolactose, it no longer binds to the operator. And when the operator is not occupied with the tetramer, then RNA polymerase can now bind and transcription can start. I hope you guys can ask a question. And that could include, would you repeat that? Has anyone seen the lac operon before? Probably some of you have, but I would guess not many.
I think I've seen it before, but I completely forgot everything I know about it.
There's so much vocabulary in this. There's a video here that goes over the lac operon, and I recommend after 2:45, it gets into stuff we don't cover. But so in biotechnology, we use the same system. This is why it's always covered in textbooks, is if you are cloning a protein and using E. coli as the factory to make it, you are using a variant of the lac operon to control when it gets made. So in biotech, we use a non-hydrolyzable analog of allolactose called IPTG as our inducer molecule. And so imagine I have my E. coli, right? And it's got its chromosome. And then what I do is I put in a plasmid. We go over this in more detail in the next chapter. And that plasmid, I can put it in the E. coli, and it's going to have the protein I want to make in it. And I'm going to have, so I would put my clone here. And if I have my E. coli just growing or being stored, I don't want it to make my cloned protein. I want to make it when the cells are happy and when I'm ready to do something with it. So normally my cloned protein would be turned off by the lac repressor. And when I'm ready to make it, and typically what I'll do is I'll start an overnight culture, and then in the morning I dilute it and give it lots of new food so that it can get what we call, it can get into what we call exponential growth. And when it's in exponential growth, it's growing fast and the cells are happy. And when I get to the beginning of that, I give it this inducer molecule, and that turns on expression of my cloned protein. And so just to put a picture on this, when I did this a ton in Vancouver, and our lab did it all the time, and we would grow our E. coli with only N15, because for the technique that I used, I needed N15 nitrogen and I needed C13 carbon. And the N15 would cost about $500 per liter, and the C13 would cost about $5,000 per liter. And so it was expensive. And we, and that's another reason why we wanted to control it, expression. And when I would grow my E. coli and I was isotope labeling it, I was always there in the lab the whole time. But there was a grad student in the lab who was a little bit more cavalier. And the C13 was the only thing the boss really cared about. He kept it in his desk. Like when you were ready to do it, you had to ask for it and he'd hand it to you. And you also had to time the induction, right? So you dilute your bacteria in the morning and it might take about 2 hours to get to exponential phase, but you never really knew. It's temperature dependent and some other things. So you had to monitor how soupy the broth looked. And then we would do it quantitatively, but then you'd give it the inducer. And Logan, the grad student in my lab, it was a Saturday and I wanted to do a hike. And so he diluted his E. coli in this expensive C13 N15 media. And we went for a hike and it took longer than we thought. And he called the lab to see if someone was in there. It was a Saturday, to see if someone was there who could check his cultures. And the boss picked up. And so he said, unfortunately, it was not my experiment, but Logan said, oh, can you check, can you measure the OD is the optical dispersion. Can you measure my bacteria? And Lawrence measured it and it was, you want to induce when it's about 0.2. And he was right at 0.2. And we were like 45 minutes away from the lab. And Lawrence said, lucky boy, and added the IPTG for him to turn on the expression of his cloned protein. So it's hard to, people always ask like, how many hours do you work in graduate school? Well, you're like, we were hiking, but he wasn't totally free. Okay, so that is the lac operon. And it turns out the lac operon is also under control of catabolite activating protein, or CAP. So the lac operon naturally is induced when the E. coli has lactose and no glucose. When both glucose and lactose are present, the cell wants to use the glucose and not the lactose, and so the lac proteins are not made. And that is accomplished through catabolite repression. So this is repression of the synthesis of the lac proteins by glucose. And the protein that does that is CAP. It binds to the promoter when complexed with cyclic AMP. And it allows RNA polymerase to bind to its entry site on the same promoter. Okay, so we have +1, the upstream region here where RNA polymerase binds, and this is the repressor binding site, that means it's downstream, and this is the CAP binding site here. Okay, so the CAP, so in this case, CAP does not bind. So in the previous case, when the inducer was there, the small molecule bound to the repressor and prevented it from binding the operator. In this case, the small molecule promotes binding. So when cyclic AMP is in complex with CAP, the protein, then it can bind to the operator. And I believe that assists the RNA polymerase for binding. So this is a cartoon illustrating the different kinds of control. You have negative control where with induction, where you have our small molecule binds the repressor and prevents it from binding. And that's with the lac repressor. But you can also have negative control where that leads to repression. And this is the trip operon has a trip repressor, but that repressor only binds in the presence of tryptophan. That's what's showing here. So in this case, the small molecule prevents binding of the repressor to the operator. Then there's positive control, which is what we just saw with CAP, where cyclic AMP binds the CAP protein, and then the CAP protein helps recruit the RNA polymerase. And this is another case here where you have an active inducer that gets, when the small molecule binds, it does not bind to the operator. The small molecule prevents operator binding, and then you don't get induction, which is repression. Am I doing on time?
Okay.
Okay. We have,
Well,
Are you gonna be home for dinner?
yeah.
oh, I think somebody needs
Sorry.
to.
Sorry.
I was just trying to find the way to mute everybody. No problem. Okay, so I'm sure you guys, your heads are spinning. So I'm gonna save the trip operon attenuator mechanism until Tuesday. Let's see. Probably the best thing to do now would be to look at some homework questions, right? Okay, that's not the one I want to look at. Oh, I gotta stop sharing this. There it is. Okay. What is the difference in the requirements for a primer in RNA transcription compared to DNA replication? Okay, so we know replication requires a primer, right? Because it needs that 3' hydroxyl. What about transcription?
Don't we not need a primer for transcription?
Correct. So there's no requirement for the 3' hydroxyl, and there are no primers in transcription. Ooh, 3 important properties in RNA polymerase from E. coli. That's kind of a general thing. Well, I would say, I'm not sure what it wanted, but I would say it goes 5' to 3'. The synthesis, it requires a template. It does not require a 3' hydroxyl. And then I would also say that make sure that you know about the core enzyme and the hollow enzyme. And the difference there is the sigma subunit. I'm just going to check to see what specifically I was looking for. Oh, that's not where I want it to be. Yeah, that's basically what I was thinking of. That was what I was, oh, and I don't know if I mentioned that it needs a template. Okay. What are the different terms to use to describe the 2 strands of DNA involved in transcription? So I would draw the duplex DNA and then list the sense and just draw a diagram with the plus and minus, coding and non-coding, template, sense and antisense. You guys want me to go over that? It's not as easy for me to do it on the screen. Right, so if I draw my DNA, right, and let me do it different just to make it. There's my duplex. Let's say that +1 is here. This is upstream. This is downstream. Which strand is going to be the template strand, the top or the bottom? Yeah. Because it's 3' is upstream. So this will be the template. Is the template plus or minus?
What do you mean by plus or minus?
It's a term used to describe whether it's the same sequence or complementary to the messenger RNA. Yeah, so the template strand, just by definition, is called the minus strand. And it's complementary to the messenger RNA, and so we call that antisense. And so this other strand, that is called the coding strand. It will be called the sense strand. And the plus strand. Okay. Promoter. There's 3 parts to the promoter: the transcription start site at +1, the -10 region, which is called the Pribnow box, and that's the TAAT region, which can open to form the open complex, and the -35 region. So this question wants you to do a sequence of nucleotides, just to show if I have a duplex, what the sequence of the messenger RNA will be. Let me get into this. Inducer and repressor. So remember, the repressor is the protein that binds to operator and it blocks transcription. An inducer is a small molecule that can bind to the repressor and cause it to dissociate from the operator, and that allows access by RNA polymerase. Okay. Are these questions helping you feel better, or are they scaring you? Oh, good. So sometimes when I have taught this stuff, I've told people to just read it and then we work on the homework, and that worked pretty well for some people, but but not enough people did the reading ahead of time, and then they were just totally, then it didn't help them. Yeah, so this stuff we'll get to. So actually this homework doesn't look terrible long, but I recommend trying to spend some time on it, just because you will get more familiar with the vocabulary, and then when we go over the trip operon, you will get you'll be able to absorb more of the material in real time, because you won't be pausing on each word and trying to figure out what it means. Okay, so any questions? I think I'm going to stop there. So I don't want to, I think you guys need to spend some time with it. I don't want to do too much of it. Okay, let me okay guys, well I'm gonna sign off. Have a good weekend.
Thank you.
Hang in there.